Management and Utilization being organized under the NAHEP CAAST of PG School IARI. Today, we have among us Dr. K.C. Bunsel, Senior Fellow, Terry Daikin Nanobiotechnology Center as our chief guest. We also have here Dr. Rashmi Agarwal, Joint Director Education and Dean IARI, and we have Dr. K.M. Manjaya, Associate Dean IARI and Nodal Officer NAHEP CAAST IARI. Welcoming and honoring guests has been an integral part of our culture. And keeping with the tradition, I would like to request Dr. Alawat to please welcome the chief guest of today, Dr. K.C. Bunsel. Sir. With the plant. Now I request you, sir, to please welcome the Joint Director Education and Dean, Dr. Rashmi Agarwal. Sir, we also welcome our Associate Dean, Dr. K.M. Manjaya. And finally, I request Dr. Veena Gupta to please welcome Dr. S.P. Alawat. Yes. Thank you, sir. Thank you, ma'am. To set the tone for further deliberations and elaborate upon this World Bank-funded National Agricultural Higher Education Project and CAAST on genomics-assisted crop improvement, I invite Dr. K.M. Manjaya for the welcome address.
Good morning to all of you. Most respected Professor K.C. Bunsel, former Director NBPGR, and Chief Guest of today's function, Dr. Rashmi Agarwal, Dean and Joint Director Education, Dr. Veena Gupta, Professor PGR and Coordinator of this training program, Dr. S.P. Alawat, Head as well as the Director of the day, other course coordinators, and former Professor Dr. Rekha Chaudhary, NAHEP core team members, the faculty of the PGR, students, the most importantly, the student trainees who have been drawn from different state agriculture universities, deemed universities, and central universities, and the faculty of other disciplines who will be contributing for the successful conduct of this training program on the topic plant genetic resources management and utilization. We have at IRI a sponsored NAHEP, that is National Agricultural Higher Education Project, under which we have two components. The one of the component, the CAAST, that is Center for Advanced Agricultural Science and Technology, in which IRI has been given the project of 20 crores to do the research as well as the HRD activities on the aspect of genomics-assisted crop improvement and management. To begin this program, or before that, I would like to apprise you, and particularly to the young friends we have come across the states and universities. Just for your information, this is a very new initiative and the only program which is more of a student-centric subcomponent of World Bank, as I just narrated. It has been monitored and guided through the Indian Council of Agricultural Research to provide a platform for strengthening educational and research activities of postgraduate and doctoral students. The IRI was selected for this prestigious program and specifically with the aim to inculcating the genomics education and skills among the students and enhancing the expertise of the faculty of IRI in the area of genomics. The IRI's CAAST project is unique as it provides a platform to the MSc and PhD students from different disciplines for learning omics skills by doing research. The students will be trained at institutions of international repute. The CAAST will also organize lectures and training programs and develop online teaching resources useful to the student and faculty of NRES. It will provide opportunities to the students and faculty to gain international exposure and also to generate and analyze big data genomics and phenomics of crops, microbes and pests for genomics augmentation of crop improvement and management. Further, the project also envisages developing a modern laboratory named as Discovery Center that will serve as a common facility for students' research at IRI. I once again welcome you all to the inauguration session of this course. The primary objective of this training program is to impart knowledge to students on various aspects of PGR, their management and utilization. At this very important occasion, it is my honor especially to profusely welcome Professor K.C. Bunsel. You may not be aware, the young students who have come other institutes or universities. He is the mastermind at the final stages of this prestigious World Bank funded project, and he could guide all of us, including all the deemed universities and the faculties, to take up this challenging task for student-centric activities. And profusely welcome Professor Bunsel for this inaugural function. And our beloved Dean, Dr. Rashmi Agarwal, who is always a student-oriented teacher and always takes a keen interest for the holistic development of all the PG school students at IRI New Delhi. And we are ably supported with, we have 26 disciplines. PGR, though it is not a part of IRI, but it is a sister institute of ICR, and they have been conducting this course. Earlier it was MSc, now they have also added PhD course with the able assistance and guidance of all the faculty members who are very senior and reputed, recognized faculty members at this very important institute. I am happy to say, ma'am, all our students really doing excellently, not only in the national level but also international level. And under this program, you would like to know that already their students have visited UK under this program. Thank you very much, ma'am, for contributing to this program. And equally important is the faculty and the students who are there in the PGR discipline. So I welcome you all, especially the students, for this inaugural function. Thank you very much.
Thank you, sir. We are very thankful for your constant support throughout. Now I request Dr. Veena Gupta, Professor PGR and Course Coordinator of this training program, to elaborate about the training.
Thank you, Ira. Good morning to all of you and our chief guest, Dr. K.C. Bunsel, Madam Dean, Dr. Rashmi Agarwal, Dr. Manjaiya, Dr. Alawa, and dear students. You all know that genetic diversity is essential to any group of organisms. If we all are alike, there might be a disease or something and everything will be swept away. So the same is true for the plants, the plant genetic resources. Their diversity is all the more important because it is used for the development of new varieties which can fulfill the demand of increasing food or for the increasing population, and it is also important to have a variety of taste. You might be having breakfast, lunch, dinner, and every time you need diversity. Single food or monotonous food will not be desirable, and also to have all the nutrients and everything in a balanced diet. So for this, diversity is important, and NBPGR is meeting the targets for collecting this diversity either through explorations or import from the other countries. And this diversity is well conserved and characterized at the National Gene Bank. And today we are proud to say that we are second in the world, having 4.5 lakh accessions conserved in the gene bank, which comprise 2,000 different species and includes the wild PD relatives, crop wild relatives, land races, traditional varieties, farmers varieties, and the germplasm. To facilitate the efficient use of this germplasm, characterization is also done for trait specific values, and then this knowledge, if it is not shared with the other partners or with the scientists or breeders, then it is of no use. So to share this knowledge and to have an efficient management of this germplasm resources which we are having at NBPGR, this training was organized under this project, which was specially organized for the students under the NAHEP system. So as the mandate of this, the training is funded from the, it is funded from the World Bank, and to our surprise and also to the surprise of our PI, Dr. Vishwanathan, who is PI of this project for us, total of 178 applications were received from 20 states, from 20 universities and 50 states of India. And it was very difficult for us to select the candidates. Then various criteria were set, and these 27 candidates which are sitting here, they are the result of those selections. So now I will request our students who have come to introduce you to the gathering. You can start from here. Sir, you can start.
I have been already, in a way, partly introduced, but my name is K.C. Bunsel. I served here for about 6 years, and currently I'm working in the Resources Institute of India, that's in New Delhi. It's a private organization, and I am a senior fellow there, working for the last about 2 years after I left NBPGR. Thank you. It's a pleasure to be here.
Thank you. Good morning, all of you. I am Dr. already introduced. I am basically a plant pathologist, alumnus of ICAR-NBPGR, and serving the institution since 1980 when I joined as a student in ICAR-NBPGR. So at present heading the division of plant pathology, and also I am with the additional charge looking after the dean and joined at division ICAR-NBPGR. Thank you.
Good morning to all. Myself, Allow. I'm head plant exploration and germplasm division. In the last 2 years, I've been coordinating the exploration and germplasm selectivity here in the NBPGR. Thank you.
माइक तो ऑन है, ये भी दोनों ऑन करके बोलो.
Good morning all. Good morning all. I am Gladys Merijoy from Kerala Agriculture University, and I am MSc final year student from Department of Plant Breeding and Genetics. So, my research title is Allelic Difference in the putative gene IPK1 and phytic acid content in black pepper.
Good morning everyone, myself Anju Alexandra. I am doing my MSc in Plant Biotechnology and I am from MACFAST, which is under Mahatma Gandhi University, Kerala.
Good morning, everyone. My name is Samir Upadhyay, and I am a PhD student from Institute of Agricultural Sciences, BHU, and I am working on QTL mapping for sodicity tolerance in real population in rice. Thank you.
Good morning to all. Myself, Rajshekhar Dwivedi, student of Genetics and Plant Breeding from Chandrasekhar Azad University, and my title is Dial Analysis in Wheat Crop for Given Parents.
Good morning, all of you. My name is Atul Pachori. I am from Indira Gandhi Agriculture University, Raipur. And my PhD title is Morphological Characterization and Molecular Analysis of BPH resistant gene in rice germ plant.
Good morning to all of you. Good morning to all of you. And my name is Manoj Avasthi from PhD Horticulture second year from Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut. And I am PhD research topic is comparative studies on the effect of rooting media, IBA concentration, and layering methods in guava.
Good morning, everyone. I am Sanket Rathi from Department Genetics Plant Breeding, Institute of Agriculture Sciences, Banaras Hindu University, Varanasi. My research topic is QTL mapping for grain quality analysis in short grain aromatic rice. Thank you.
Good morning, everyone. My name is Sushant Bornak and I am from Kolhapur, Maharashtra. Right now I'm pursuing MPhil in Shivaji University. My research topic is systematic study of order agaricales from Kolhapur district, which includes the identification of gill mushrooms. Thank you.
Good morning, everyone. I am Siddhant Rajan Padi and I am MSc final student from Division of PGR in IRI. And my current research topic is development of core and reference set of copy using biochemical traits. Thank you.
Good morning, everyone. Myself, Mallikarjun Brother, in first year MSc student in the Division of Plant Genetic Resource from IRI, New Delhi.
Good morning, everyone. Myself, Suma. I'm doing my senior MSc in Department of Genetics and Plant Breeding from University of Agricultural Sciences, GKVK, Bangalore. My research is genetics of Persicum leaf blight disease resistance in maize. Thank you.
Good morning, everyone. Myself, Soumya. I am from University of Agricultural Sciences, Dharwad, from Department of Genetics and Plant Breeding. I am third year PhD student and my research topic is on genetic studies for drought tolerance in groundnut.
Good morning, everyone. Myself, Manmohan Singh, in Genetics and Plant Breeding Department in Rajasthan College of Agriculture under MPUIT Udaipur. Thank you.
My name is Sandeep Kumar Bangadwa from MPUT Udaipur, PhD fifth semester student. My research work is on stability analysis in baby corn.
Good morning, everyone. Myself, Shilpi Pachori. Currently, I am pursuing my master's in plant breeding and genetics from JNKVP, Jabalpur. And my research topic is molecular characterization and physiological studies on soybean germplasm.
Good morning to all. Myself, Shikha Upadhyay. I am from JNKV Jabalpur campus. I am pursuing PhD previous year student.
Good morning to all of you. I am Rinki Arya from Dr. YSP UHF Noni, Solan, Himachal Pradesh, and I am PhD third year student. I am doing my research on the effect of insect pest management modules on seed quality and yield parameters of capsicum annum. Thank you.
Good morning, everyone. I'm Khonang Longko from Assam Agriculture University. I'm a PhD scholar in the Department of Plant Breeding and Genetics, and my research topic is QTL mapping for drought tolerance in rice.
Good morning to all of you. I am Saeed Tawkir Ali, currently pursuing a Masters in Biotechnology from Delhi Technological University.
Good morning to all. Myself, Manikumar, from Andhra Pradesh. Presently, I am a PhD scholar in Tamil Nadu Agriculture University. I am working on improvement of Gundu chili grown under rainfed condition for yield and quality. Thank you.
Good morning, all. Myself, Ashish Kumar Swai, from Central University of Jharkhand. I am pursuing my MSc Life Science in Central University of Jharkhand.
Good morning to everyone. I am Sushmita Seni and I am pursuing my MSc second year in Biotechnology from School of Agricultural Biotechnology, Punjab Agriculture University, Ludhiana. And my research title is Whole Transcriptome Assembly and Annotation of Agelops speltoides for identification of heat stress tolerance genes. Thank you.
Good morning to all. My name is Goti Hirvita. I am come from Navsari Agriculture University, Gujarat, and student of plant breeding and genetics. And my research topics is interaction and molecular biology in rainfall pigeon bee.
Hello everybody. I am Rekha Chaudhary. I am working on cryopreservation at NBPGR New Delhi.
Good morning. Myself Dr. Veena Gupta. I am Professor PGR at NBPGR.
Once again, good morning to all of you. Myself Dr. K.M. Manjaya, Principal Scientist in the discipline of Soil Science and Agriculture Chemistry, Associate Dean PG School IRI, and Nodal Officer for this CAAST program at IRI. Thank
you. Good morning, all of you. I am Ashok Kumar. Basically, I am plant breeder. I am working the characterization and evaluation at NBPGR since last 30 years. And presently, I am heading the division of germplasm evaluation and also coordinating the evaluation activity at national level. Thank you.
Good morning. I am Kavita Gupta. I am Principal Scientist in the Division of Plant Quarantine.
Good morning, everybody. I'm a principal scientist in Division of Tissue Culture and Cryopreservation, and I'm working on in vitro conservation and cryopreservation of bulbous and ornamental crops. Thank you. Ruchira Pandey. I don't know whether I told my name. I'm Ruchira.
Good morning, everyone. I am Sandhya Gupta, Principal Scientist in Tissue Culture and Cryopreservation. I am working on in vitro conservation and cryopreservation of temperate fruit crops. Thank you.
Good morning everyone, myself Shubham Dev. Currently I am pursuing MSc first year in the Division of Plant Genetic Resources, IRI New Delhi.
Good morning everyone, myself Sandeep Kumar Paningrai. Currently I am pursuing MSc first year in IRI Division of Plant Genetic Resources at IRI New Delhi.
Good morning, I am Praveen Gumchanwadi, first year MSc students, Division of Plant Genetic Resources, IRI New Delhi.
I am Nagaraj Nagdi, Plant Genetic Resource, IRI New Delhi.
Good morning everyone, my name is Abhishek. I am MSc second year student. I am working on evaluation of diverse copy germplasm against bean common mosaic virus and division of PGR IRI.
Good morning to all. I am Dr. K.C. Bhatt from Plant Expression Division of NBPGR. I involved in research and teaching both.
Good morning to everyone. I am Asutosh Kumar from DRPSU Pusa, Bihar, PhD final year student. Presently, I am working on genetic aspects for zinc biofortification in rice, and I am from plant breeding and genetics. Thank you.
Good morning, everyone. Myself, Venugopal Gowda. I am a senior MSc, Division of Plant Genetic Resources. My research topic on molecular cytogenetics on amaranthus.
Good morning, everyone. I am Chetan Kumar, second year MSc student, Division of PGR, IRI New Delhi. My research problem is assessment of differential reaction of copy accessions against brucids and diversity analysis.
Good morning, everybody. I am Anuradha Agrawal, Principal Scientist in the Tissue Culture and Cryopreservation Unit, and also taking care of the unit as Officer in Charge.
I am Anjali Kak. I am Principal Scientist in the Division of Germplasm Conservation.
I am Sherry Rachel Jacob, working in the National Gene Bank, looking after conservation of cereal crops.
I'm Mukesh Kumar Rana, Principal Scientist in the Division of Genomic Resources and looking after DNA fingerprinting of crop varieties.
Good morning, everyone. I'm Lalitarya, working as Principal Scientist in the Division of Genomic Resources and basically working in genomics of small millets. Thank you.
Good morning, everyone. I'm Dr. Vartika Srivastav, and I'm a scientist in Tissue Culture and Cryopreservation Unit.
Good morning, everyone. I am Gautami, scientist in Tissue Culture and Cryopreservation Unit.
Good morning, all.
I'm Anjula Pandey, and also I'm in the exploration division and taking care of the faculty. I'm also the faculty member of plant taxonomy, teaching 2 courses here.
Good morning all. I am Kuldeep Tripathi, scientist in Division of Germplasm Evaluation. I am looking after evaluation of pulses germplasm.
Good morning everyone. I am Dr. Bharat, working as a nematologist in the plant quarantine division of NBPGR.
Good morning. I am Sunil Archak. I am ICR National Fellow at NBPGR, and I work on PGR informatics. I'll be interacting with you on this particular task aspect tomorrow.
Good morning everyone. I am Celia Chalam, Principal Scientist, Division of Plant Quarantine. I am working on quarantine of germplasm including transgenics for viruses especially, and screening of germplasm against viruses at phytoplasma. And I am a faculty of PGR and I teach plant quarantine. Thank you.
Good morning. I am Vandana Tyagi and I am a Principal Scientist in Germplasm Exchange and Policy Unit.
Good morning. I am Pratibha Brahmi. I am Principal Scientist and in charge of the Germplasm Exchange and Policy Unit. Thank you.
Good morning. Myself, Sangeeta Bunsel. I am working as Principal Scientist in the Tissue Culture and Cryopreservation Unit and looking after the in vitro conservation and cryopreservation of tuber crops. Thank you.
Good morning, I'm Ira Malhotra. I'm a scientist in the Tissue Culture and Cryopreservation Unit looking after tissue culture and cryopreservation of spices crops. And thank you each one of you. You are all the lifelines of this training program. And now may I request Dr. Rashmi Agarwal, Joint Director Education and Dean, for her comments, please.
Once again, good morning to all of you. Honorable Chief Guest of today's function, Dr. K.C. Bunsel, who was also ex-director of NBPGR, at present Senior Fellow at TERI. Our Associate Dean, Dr. K.M. Manjaiya, Dr. Veena Gupta, who is the course coordinator, also the professor of PGR. I'll hold. Dr. Elawat, Dr. Veena Gupta, faculty of NBPGR, students who are, I think, the students of PGR, and also the student trainees who have come here, and other staff of NBPGR, ladies and gentlemen. It's really a great pleasure to be here amongst you today morning on this inaugural of training program, as very rightly said by Dr. K.M. Manjaiya. This is a very prestigious project which IRA has got by World Bank ICR together funded program, the CAAST program, Center for Advanced Science and Technology under NAHEP project. It's a multi-crore project wherein he said that it is student-centric. We are training the students across the nation on different aspects. And today's aspect, as said, plant genetic resources and their management. I see among the students lot of diversity. As Veena also said, there is lot of diversity in the genetic resources. Among the students, I find some of the students are in the mushroom also, some are in the crop protection, IPM, and others were working on various crops, starting from rice, wheat, pigeon pea, and many others, soya bean, capsicum, chili, lot of crops students, they have chosen their topic on. So this program, I think the course director is our director of NBPGR and other course coordinators like Veena Gupta and Ira. They have structured this program in a very nice manner for 10 days, during which you will be exposed to all the lectures and also hands-on training program, how you characterize, how you evaluate, and how you can utilize. See, characterization, evaluation, exchange, import, export, everything is fine. It's all you can learn. But how you utilize these plant genetic resources to a useful purpose, that's very important. That's where the gene mining or the trait identification comes and plays an important role. One of the students said she's or he, I think, working on Agilops, speltoides. You know, it's a very important source for many of the genes, which can be utilized for transfer of the genes through regular breeding program or other breeding programs. So this plant genetic resources finds a very, very important role in each discipline, across the be it entomology, pathology, or any other discipline. Of course, genetics and plant breeding, I find most of the students are from genetics and plant breeding. So this exposure at this young age, because at this age, all of you, your minds are very fertile. Whatever gets imprinted it will be really very useful. So at this young age, if you are trained, if you are given, then you can utilize this training program for all your future endeavors. Some of you are MSc students, so you can utilize this knowledge for your PhD programs. And the PhD students can further will be joining somewhere as scientists, maybe in universities or in ARS system. So this training will be very, very useful to you. Not only this training program which is being imparted here, you will also be interacting with each other. So this is your first interactive, like you have come from many different universities, as said 15 different states and maybe how many? 20 different universities. So it's an opportunity for you to interact with each other, develop good relationships, which will really be nurtured in many more years in future. And during the program, I feel I have seen that you will be visiting many biodiversity parks also. Like you may be aware of it, or if you are not aware, you will be visiting Yamuna Biodiversity Park is there. It's developed by, I think, Delhi University. Yeah, DDA and Delhi University. We keep hearing, you must have also read many news in the newspapers also about it. And also ecological restoration. This is another इसी में ही है. Okay, this is in the same, because I've not visited as such. I think I should get a chance to visit. So that's a really useful visits. You may also visit the state-of-the-art facilities like phenomics in IRI. So how you can phenotype without destroying the plants. So in situ phenomics facilities there, which you will be visiting, and many other facilities may be in the pipeline you may be visiting. In NBPGR also there are a lot of facilities, as you have heard the faculty, cryopreservation, gene bank, and genomics laboratories. So maybe hands-on training little bit on how bioinformatics also may be a part of your training program. So how this recent full genome sequence which we have generated for a large number of crops in nationally, also internationally, and also for the microbes, how you can utilize this genomics or the whole genome sequence data for useful purposes, for identifying the markers, identifying the QTLs, or any other useful genes which can be utilized for future characterization and for improving the crop plants. So I'm really very happy that 27 students have been selected. In fact, our program was maximum to select 25. So you have gone ahead, because maybe you received a lot of applications. 178 applications is a very good number received for this particular course. And as I said, it's a basis of agriculture. So across the disciplines, PGR is very, very important. So with these words, I welcome all of you on this training program on behalf of PG School IRI. And I'm very grateful that, honored that NBPGR invited me on this occasion, and all the best wishes to all the students. Thank you very much.
Thank you, ma'am, for your kind words, and also for sparing time to come and, from your busy schedule, to come here and grace the occasion. Moving on, now I would like to introduce our chief guest, Professor K.C. Bunsel. Professor K.C. Bunsel, Senior Fellow at Terry Daikin Nanobiotechnology Center, Gurgaon, and also the former Director of the National Bureau of Plant Genetic Resources, is an accomplished scientist of international repute. Having obtained his PhD with gold medal from IARI in 1989, he has pursued advanced research from Harvard University, Cambridge, USA. He has occupied eminent positions in ICAR, such as the Principal Scientist in Plant Biotechnology, Professor from the year 2004 to 10 at IARI. He was also the coordinator of the National Project on Transgenic Crops from 2006 to 10, and then the director of NBPGR from 2010 to 16. He has to his credit over 150 research papers, review articles, and book chapters, and his research findings and outcomes are cited in crop improvement programs in several laboratories within as well as outside the country. Professor Bunsel is a recipient of several awards, both at the national as well as international levels. To cite a few, he has been awarded the highest award of ICR for an individual's contribution, that is the Rafi Ahmed Kidwai Award. He has been awarded by the Indian Science Congress, which was conferred upon him by the Honorable Prime Minister of India. He also has been awarded the Biotech Career Award of the Rockefeller Foundation of USA. He has to his credit a recognition award of the National Academy of Agricultural Sciences and also the prestigious Haryana Vigyan Ratan, which were conferred to him by the Honorable Governor and Chief Minister of Haryana. Professor Bunsel was also elected as the Vice Chair from Asia for the 15th regular session of the Commission of Genetic Resources for Food and Agriculture of the United Nations from the year 2013 to 15. He is also a recognized popular science teacher and a research guide. And as director of NBPGR, Professor Bunsel was conferred upon the Research Leadership Award for his outstanding contributions in research coordination and management. Sir, it is an honor for us to have you here. May I please invite you to deliver the inaugural address? Yes.
Well, friends, good afternoon to all of you. Rather, good morning, not afternoon yet. Good morning to all of you. Acting Director of this institute, Dr. Ahlawat, Joint Director and Dean, IRI, Dr. Rashmi Agarwal, the Associate Dean, Dr. Manjaya from IRI, Dr. Veena Gupta, the coordinator of this particular program and head conservation division of this institute, and the professor also of the PG school of the subject of plant genetic resources, former professor Dr. Rekha, very senior faculty members, scientists of this great institution, National Bureau of Plant Genetic Resources, the staff of this institute, and you, the dear participants, the students, from various states and various universities who have gathered here for this particular training program, and last but not the least, the students of the PGR fraternity here from NBPGR. It's an honor for me to be joining you this morning, and I thank actually the director of this institute, Dr. Veena Gupta, Dr. Ira Malhotra, for inviting me once again to this institute, which I always love to come back again and again. It's a kind of homecoming for me. NBPGR, I'm sure you all would know more, is a great institution. And actually, I'm very happy today that a program, which is National Agricultural Higher Education Project, the World Bank sponsored and sponsored by Government of India, is getting implemented by the ICAR. It's a very important program. And I know how much hard work people have put into it. It took about 4 to 5 years of the efforts alone for preparing, formulating this project and getting it through. And finally getting approved by not only World Bank, but also Government of India. It was a very major task. And I must, at this juncture, actually congratulate and remember the contribution and the leadership given by our Honorable Director General, Dr. Tirlochan Mahapatra. It was his vision, it was his actually speaking a kind of a direction to me that this is the project. We must try to bring it to the country. For the first time ever, any World Bank sponsored project has come to India related with higher agricultural education. And I'm very happy about it, that I think this is again, to me and my knowledge, is the first time that the students, masters and PhD students, are being trained in different subject matters. Particularly, I'm talking of the management of plant genetic resources. While plant genetic resources remain the backbone, the fundamental, kind of material you use always for any kind of science you're talking about it, particularly related to agriculture, crop production, etc. But very many people, actually speaking, particularly students, have never been exposed to the importance at the national or international level of plant genetic resources. And And if you want to know more about plant genetic resources, the only institution in the country, okay, which is of international fame, is this institute, the National Bureau of Plant Genetic Resources. Among 100 and odd institutions we have in ICAR, this is one institution on plant genetic resources with regard to crops. Of course, there are others like, fish genetic resources, you've got animal genetic resources, you've got insect genetic resources, you've got microbial genetic resources also there. But with regard to the crop production, this is the institute wherein you must be always in constant touch with for one thing or the other, as has been mentioned very clearly by Dr. Rashmi Agarwal and also the other speakers, that how important is the knowledge, first of all, you must have about these plant genetic resources. And just to give you, with the available time I have, a holistic viewpoint, actually speaking, while every aspect is being touched here, will be carried forward, to this manual you have through this next 10 days of training. Dr. Veena Gupta mentioned about the importance of various disciplines of plant genetic resources, kind of exposure you will be having it. But I thought to give you a holistic viewpoint is this, that, in this whole world, the most important organisms are all living biological resources, all living biological resources. You talk about it, all which are living in nature, they're all biological resources. And which we cover it as called in general as biodiversity. But coming to agriculture, we specifically talk about agrobiodiversity, where we're talking about all the genetic resources, not only plants, but even others, including livestock, animal genetic resources. We have a bureau in Karnal, and others I mentioned before. And the whole world is recognizing the importance of it even now more and more, of these kind of agrobiodiversity which is available, or biodiversity in general which is available on this planet Earth. And then it has gone to the extent that there are several now international instruments, actually speaking, to manage these biological resources in the world by different nations to which they belong to. From 1993 onward, any country will have a sovereign right on all these biological resources which are present and found or occur in their own piece of land. Like in India, we have whatever the germplasm, whatever the biological resources, we are having the sovereign right of it in terms of legally speaking, by this instrument we call it as Convention on Biological Diversity. Okay, and utilization of it is also being promoted further, more and more. It has been utilized in the past. I'll give you a little bit of historical perspective of that as well with regard to crop production. But then this is to be utilized more and more, and also to reward those farmers or anybody for that matter who was associated with the management of this, knowingly or unknowingly, what value this kind of material will carry with them, or with this material for times to come. So there are, some sort of associated kind of benefit sharing instruments, national, international, associated with these programs. And there is an international treaty also further to promote the exchange of germplasm across the globe. Before 1993, it was free for all. Several people actually, institutions, including our international research institutions, are boasting on the kind of material which they have taken for research, breeding, training, or for production of high-yielding varieties from India, from this very national gene bank, which is the second largest gene bank in the world after USA in terms of number of accessions conserved here as ex situ conservation in the national gene bank, like NBPGR. So, but then, these natural resources, they all occur in their natural habitats, right? They are exposed to various evolutionary changes which is taking place as a result of changing in weather and the climate, which is very important, right? So they are there from where actually our scientists from NBPGR, they've been collecting it or preserving it for you and bringing it here, doing all, kind of processing which is required. But also the ones which are conserved, we call them as in situ or on farm. Also they are there, which were gathered by the farmers, and it was actually the farmers who started selecting or finding the best out of what was available in natural habitats, for their own domestication purposes, for their own kind of having the food for them to survive and eat. And it all started about 10 to 12,000 years ago when we started talking of agriculture, right? So that's one side of it, actually speaking. But the other side of it is that sometimes, the science now came into picture, okay? That we need to produce more because, the population has always been exploding, So then the scientists became concerned and, whatever the role farmers played, fine, but now we need to bring science into it, actually speaking. The whole gamut of, you talk of management of plant genetic resources or utilization of plant genetic resources or producing those high-yielding varieties, utilizing these plant genetic resources. So it was, again, if you talk of historically in 1860, I believe, that the Mendel's laws, or Mendel gave those laws, which remain hidden until they were rediscovered in 1905, almost about a half a century later. But after that, actually, based on those principles of genetics, the tools were developed to improve these crop plants or utilization, to improve the utilization, I would say, of these plant genetic resources for breeding high-yielding varieties, and through the process of, we call it as plant breeding. And until 1950, there was not much progress to increase the production or increase the productivity, actually speaking, of a particular crop variety beyond, let's say, 1 ton per hectare. But it was again the science of genetics and the tools of plant breeding were utilized effectively, through a Rockefeller program by Dr. Norman Borlaug and his team, particularly with regard to wheat. And it was then in 1960 to 70 that we witnessed the benefits of science, or the benefits of science of genetics and the tools of plant breeding, that the production could go up to, now, let's say 3 to 5 tons per hectare against 1 ton per hectare. So we developed those high-yielding varieties. So that was very important on one side of it. You must know that. It was required to feed our population. I don't want to go into again, another historical background of it, that people were dying out of hunger, out of starvation, in mid-19th century. We didn't have enough, not to get and buy it, we didn't have enough as an excess of that food which was available. People died out of hunger, So anyway, this Green Revolution was a welcome step, was important, but then it also had, in a way, if I may mention so, a kind of negative effect, I will say, on biodiversity or agrobiodiversity, which was being saved or conserved on farm, in situ, by our millions of farmers. Particularly, I'm talking not only in India, even elsewhere, wherever Green Revolution touched, for example, maize hybrids or soybean production, high-yielding varieties in US and many other countries around the world. Any farmer who was growing, let's say, at least hundreds of those different farmers' varieties, you call them, or obsolete varieties, which were otherwise selected by themselves out of the material which they collected from time to time, those actually disappeared from their fields. And it was bound to happen because India wanted to produce more. Against 50 million tons, which we had roughly, let's say, 1950, we wanted 200 million tons. Today we want 300 million tons. We already touched about 285 million tons, So for that, what do you need? You need these high-yielding varieties, okay? But unfortunately, there was a dilemma now, kind of, that what will happen to those varieties which were, over the years, 10,000 years or so, were there, kind of material, which was subjected to all those evolutionary, pressures. And that was conserved here, actually was collected. And that's a great effort, the scientists ever, starting from Dr. Harbhajan Singh, actually, who was the first explorer, the collection he did under, the single scientist actually whose name we are celebrating and sitting here in this particular Harbhajan, hall in this NBPGR. So those all varieties, by the great efforts of all scientists, were collected, conserved, and that became the backbone of further, increasing production, right? So that was one thing, or this another point. Then on the other hand, what was happening in 1953, first time structure of DNA was discovered, right? We did not know, really speaking, when we had these varieties, or farmers or even scientists were using, making crosses, that what was that which is getting inherited generation after generation? What is that principle, they used to call it, or DNA, which is carrying this, good information or whatever information it carries from generation to generation? Or what are those genes, really speaking, governing these different traits? right? So in 1953, this DNA was discovered, structure was discovered. Then later, a lot of science, we started having it in this area, of molecular science. 1970s, cloning was discovered, that you could take a gene from elsewhere, from any organism species. You can isolate it, in a, let's say, a test tube, and you can clone it. You can have it conserved in your lab at that particular gene. In 1980s, the methods were discovered to transfer the gene from one species to another. We never had an ability to transfer the genes, let's say, from bacteria to plants. It was in early 90s, 1983, that this method was discovered, by the scientists. And today, those scientists got the Nobel or the World Food Prize, actually speaking. 3 scientists, if I remember, Meridel Chilton, Robert Horsch from Monsanto, and Mark von Montagu from Belgium. These were the scientists. So then the era started of GM crops, right? And that's even continuing with the GM crops. The idea here was that you can take any gene from bacteria or any other source and you can transfer it to the plants, improve the crop plants. So from these obsolete varieties, farmers' varieties, from two high-yielding varieties, we came down to these GM crops with many other traits which we did not have the benefit of getting them transformed from existing plant genetic resources, right? But then, simultaneously, let's say sometimes in 2000, genomics came into being, in the sense that scientists discovered how to sequence the whole genome, how to first of all sequence a stretch of DNA, for example, a method, or few methods were developed. I have had the honor of actually working with one of them who was head of the department, Wally Gilbert, who got the Nobel Prize for it in 1977, to give a method of sequencing of this DNA. So therefore, sometimes in 2000, scientists started talking of sequencing the whole genome of a particular crop plant or a plant, tiny plant or Rhodopsis we started with, came to rice, came to, in fact, wheat now, which was a very Herculean task, but in between, a lot of crop plants were sequenced as a whole genome, right? Tomato, potato, and many other fruit plants, papaya, grapes, you talk about it, and several of them, as a single one of the varieties out of all these, material which was available was sequenced as a whole crop genome, okay? And that became a reference genome sequence. Idea here is what? Idea is to look for those genes which are important, could be associated with a particular trait. But before one could do this, now idea was again to accelerate the pace of that plant breeding, conventional plant breeding. Instead of having phenotypic selection based on morphological features, can you do it based on a piece of DNA present on the genome of that particular plant species or a variety, So that molecular breeding came into being on the other hand. Because of the availability of these tools were so strong that time, were so important that several varieties in India, also of course outside India, were developed to start with rice, bacterial leaf blight, etc., pearl millet, many other crops based on molecular markers or marker selection. To see the presence or kind of an absence of a particular gene of interest in a particular given variety. But then the tools of genomics further were developed more and more in the sense that the cost came down drastically of sequencing. And then today, scientists are talking of sequencing thousands of crop varieties, for example. Okay? And then also now scientists are rediscovering the role these gene banks are playing. While there has always been a concern that the gene bank material is not utilized fully to the extent that it should have been utilized, if you talk of the total availability of the germplasm across the world and what percentage of it is utilized in crop improvement by breeders, is about 1%. So you can imagine how much information still waiting to be discovered on genes and alleles which are important And you have the material available with you in gene banks like this across the world. So then, idea was that can we evaluate, characterize more and more material by growing them in the field, or finding any new trait which is associated for increasing the production, or making plants resistant to a particular disease or insects. And not that you will find everything in the germplasm. There are certain crops, we have got excellent germplasm in the sense thousands of accessions are conserved, but you don't have that particular single trait which you need to be incorporated any high-yielding variety through conventional breeding. So then, of course, you fall back again on the route of genetic transformation and you develop GM crops. BT cotton is one such example. BT brinjal, while people do claim that there are species, wild species of brinjal where there is an insect resistance available, but also remember, it's not easy again to transfer from even one crop species to another because they may not be compatible enough to be crossed, to be hybridized. Or even if they are successfully hybridized, will you be able to get that fertile seed, which will give you a next generation of plant? So there are methods, of course, to take forward that, but the people thought easiest way would be to even isolate a gene from that very species, and using method of genetic transformation, you can develop a new variety. But then, science is always progressing. You won't imagine at what pace it's progressing at a great space, it's always progressing. Then the discovery came of this CRISPR. I don't know, I'm sure all of you would know it. At least in my lifetime, this is the only technology which I have seen spreading it so fast. Though we used to take a pride in talking ourselves about genetic engineering, about many other methods of isolating genes from any different species, and irrespective of crossing that barrier, you were able to transfer it. And of course, there is always, kind of another school of thought that we should not be doing this for various reasons. I'm not going to go into the details of that. But then again, on one hand now, you have these thousands and accessions of different crop species conserved. I'm sure NBPGR must be touching the number of close to 4.5 lakhs, which is 0.445 million, which is again the second largest, I said, in the world, and then on one hand you have this material, on the other hand you have these methods of sequencing, and people are doing it, utilizing these sequencing tools to decipher or find out not only what those genes are, even to find out the very biological basis of crop production, increasing crop production, understanding the various physiological processes, etc. So now we are at a stage where on one hand, thousands of these genomes are sequenced, gene bank genomics, we call it, is being done. On the other hand, you have tools of this CRISPR technology. We call it a genome engineering technology, right? So if you don't want to take a gene of interest from one species and transfer it to a plant, let's say from bacteria to a plant, or from a fish to a plant, or from an elephant to a plant, or from a human being to a plant, if you don't want to do that, suppose. Now here there is a technology available to you to explore all those natural genetic variation which is existing in these thousands of germplasm accessions, to create that kind of a variation in a given gene in a very precise manner, in a high-yielding variety, to create a new variety altogether through the process of genome engineering. So anyway, what I'm trying to tell you here is this, that the plant's own genome can be now can be engineered, okay, or in older parlance we call it can be mutated, which the scientists actually speaking, also did use as one of the tools through radiation, through chemicals, etc. But here, if you want to now duplicate a kind of a genetic variation which you have discovered in one particular accession, which is otherwise not very high yielding, that particular accession, but has that particular variation, can you now create into another variety of wheat, for example? Yes, you can do it precisely, without addition of any extraneous gene from another source. Excuse me. So, this is the whole perspective, I thought. I put it before you. Science is ever-changing field. You come from different disciplines, and I'm sure you will not be able to practice all what you want to in a given time frame of 3 to 5 years of PhD, but as a scientist, I'm sure you have a long way to go, and to remember that here's the germplasm, here are these lines which remain hidden in terms of genetic potential or otherwise in a gene bank with ex situ collection, or also not forgetting that there's a lot of germplasm which is present out in the farmer's field, in situ, on farm, or even in natural habitats, which still need to be discovered and collected and find value into it. There's a lot. So you have to come out of those silos, really speaking, if I may say so, of your thesis topics, which must have been given to you, I can understand, with all the intelligent framework or mind into it to decide that particular problem. But what I'm trying to say is that there's a lot which remains to be discovered, and finding value from these unexplored kind of germplasm you have. Technologies of tools and science must be utilized. Days are going to be forgotten, I must tell you, that you will characterize and evaluate this material anymore using morphological features by groom them into the field. My friends here in NBPGR, I'm sure, would know better than me. We are going to have a kind of a war. It occurs to me like this, that what will be the fate of these physically available with us, these plant genetic resources, versus in this digital world, the gene sequence which is available with you, which can be synthesized. Synthetic biology is coming in a big way. So are we really going to worry more and more about these plant genetic resources? To me, personally speaking, it will always remain a wealth, invaluable wealth. Okay, whole international scientific fraternity is into it, is recognizing it. But at the same time, you have many other players who may like to take the benefit of this sequencing technology in a manner that this information which is available as a digital sequence information, is enough for them rather than having the physical access to this germplasm and utilizing it in any form of crop improvement. So that's also, in fact, kind of a struggle or a debate is going on actually at the world level. That what's that we are going to do in future with regard to promoting the utilization. Actually, I've been an advocate of, I must tell you clearly, promoting more and more utilization of the germplasm for crop improvement by our own Indian scientists, by our own Indian plant breeders. Okay, and we as the scientists here, in the system at NBPGR, I'm sure all of them are playing a big role. And it's not easy again. Also the scientists or the students or budding scientists, if I may so, those who have gathered here, must appreciate, okay, when you go back to your institutions, kind of efforts the scientists of these institutions must have been doing it for the last, if not more, 100 years, if you talk the history of, collection of the material right from the inception of, this Indian Agricultural Research Institute, and all that. This is very important. So a lot of efforts is going into it. Okay. So also what I wonder, that only 27 students, only 27 is not really a good idea. Okay, it's a good idea to have it given time, 27 students to be trained. But I would suggest to Dr. Manjaya or to the Honorable National Director and National Coordinators of NAHEP, the idea is to spread this knowledge across to each and every student. You have to find ways and means in this communication world, where we have all that which is available. Why couldn't it spread to all students who require this kind of a training? It's not like a school children, that you have only to admit 100 school children, they don't care about other lakhs of them. No, I think NAHEP program must address to the needs with regard to, I'm talking of the requirement based, as we have got a training here organized on PGR management. It must spread to each and every student working in the area of agriculture, crop production or fish production or animal or livestock or to anyone for that matter. Even if they need to organize, this is the first training course, for example, second or third or fourth, like this, it should be done. It won't cost you, really speaking, much. We can find other means, that it can be, spread over to all of the universities, to all those students who could not unfortunately come here physically or couldn't be selected. So then only the whole purpose of this first ever project we have launched here in this country on national higher education will really become a fruitful activity, So that should be the goal, and I'm sure I will, of course, request personally to the national director, who fortunately happens to be one of our own scientists from NBPGR, by the way, Dr. R.C. Agarwal today, and there are very many good national coordinators who are working in different fields of science. They're manning that particular project at the moment. So my appeal to the officials would be, and also to this fraternity of NBPGR scientists, that how can you take this training forward through more number of students. I'm talking particularly of PGR. I'm not talking of any other subject of science, while genomics and all is very extremely important. The very basic knowledge of PGR, the management, I give you a little bit of holistic historical viewpoint of it, is very important, should be taken forward, by each and every student. And not only as a knowledge, when you go back, you must also be, I think, in a way, disseminating this knowledge, in your own department, by giving a small piece of seminar, or kind of information, whatever you think you would have gathered here in 10 days of time, and send that report back to Dr. Veena Gupta, with a copy to Dr. Rashmi Agarwal, or to Dr. Manjaiya, or to Dr. Vishwanath, who is the PI of this project. Under the aegis of this project, if this particular training is organized, it should be the way that you then send a report that, yes, so many students, or even scientists for that matter, you invite even your head of the department, give a seminar, and send this report back here for the kind of knowledge, to enrich themselves again, how far this training must have spread this message, particularly on the science of PGR, plant genetic resources, and particularly also highlighting the tools and technologies which are now available, and how much wealth we have in our country. We keep boasting of second largest gene bank, but how much are we utilizing it? How much we have got this benefit, as much as we got in terms of green revolution by utilization of these wheat genetic resources from different countries. But for those wheat genetic resources, we would not have witnessed green revolution at all. So what is that kind of a jump ever we have come across, based on still very important tools of plant breeding science and technology? It's important how many can we take it forward that way. So I think with these words, I say only at the end that how pleased I am to be here. I thank the NAHEP, and I thank NBPGR, and also, of course, more importantly, I thank IRI, for organizing this particular training course for the benefit of the master's and PhD students. And at least to my own satisfaction, I'm very happy that this has been organized, because it gives me immense pleasure when I come to know that this program, for which we really struggled hard, I must tell you, has come to this stage, is being implemented fruitfully, and this message is going around to all 75 state agriculture universities, and also to the deemed universities and central universities, and to various institutions of ICAR. And I'm sure international fraternity would also keep a watch, particularly the World Bank, really speaking, that how much difference are we making, through this kind of 1,100 crores of rupees which we have in this particular project. With this, thank you very much.
Thank you, sir, for the interesting, engrossing, and inspiring talk. Now, I would like to invite Dr. S.P. Alawat to please say a few words.
Morning to all. Respected Professor K.C. Bunsel, Chief Guest of today's function, Senior Fellow Terry, and the former Director NBPGR, Dr. Rashmi Agarwal, Joint Director Education, Dean IRI, Dr. K.M. Manjaiya, my colleague, Head of the Divisions in NBPGR, Dr. Veena Gupta, Course Director of this program, and others, my colleague scientists or officer in charge, and dear students, I am very pleased to be amongst with you And the part of this training, this program, today's function program, then first of all, I would like to thank Professor K.C. Bunsel for his vision to start this program, because and as well as we all know that how much pain he has taken the last time to getting it materialized. And I also thank to Dr. Rasmi Agrawal and K. Manjuya, Joint Director, Dean IRI, to initiating, submitting this project also. And as well as including NBPGR as a course, including this course in that program. We must thank to you, madam, for that also. And Dr. I would like to thank Professor K.C. Bunsel especially for giving this very comprehensive lecture, as well as starting from the historical perspective to bringing up to the CRISPR technology, as well as putting all the facts of the Bureau, PGR, as well as highlighting their importance and motivating to the scientists, to the students also, what are the problems which they can take up, what are the challenges before us which they can address. So many things Dr. Bunsel has explained, starting in situ, on farm, so many recent techniques, and so I must thank you, sir, giving such an excellent lecture. And I know you are already very popular professor, have got the leading scientist research award. So that's all because of your such qualities. And whenever we heard you, that's always very encouraging to us that your lectures also very specific, tailored. You make them. And today also, you as a chief guest, you have given all the background of the PGR to the students, which they will be coming in this time. Thank you very much, sir. Now, Dr. Rasmi Agrawal, thanks a lot for this coming this program and taking your especially valuable time. To Dr. K. Manjuya also. Then coming to the another point, then to the students, I also congratulate you that among 178 students, you have 27, you have got the opportunity. And this, as Dr. Saab has told just now, that this was not the opportunity available earlier to in the universities and other places, such kind of training. This is a very specialized training program, particularly for the students level. So this first, you are very fortunate for that, and I congratulate you for coming this. This is a first time initiative has been taken. Next, coming to the this program, this 12 days program, which it would not be possible for us to give you in-depth knowledge, because the scientists dealing here giving lecture, they have very high expertise in that. In fact, it is a, Dr. Saab has already told that it is a internationally recognized center of excellence in the PGR training. And the scientists, you know, they will be giving you the lectures, they have very high, but it won't be possible for them to give you the details of the things. But that will give you give you some ideas, things, what you can pursue, about which you can take up, because this PGR involves two very broad disciplines, the botany as well as agriculture. In botany, you can see it starts from the evolutionary process, then ecogeography, then the recently remote sensing will also come in between that. Then I would like to see technology also part of the agriculture from this, that madam will be conservation, so many things will be teaching you. Then in terms of the agriculture, you will came to the evaluation, Dr. Saab has already told, evaluation, characterization, biotic, abiotic stress, phenomics, madam has told, these things you will be, then now the pre-breeding has also come into the picture, that will, they will be able to tell you some brief just the things about all those things. Then disease and pest, quarantine must be there. So it's all, so many things you will, the policies also will be come in that picture. So many things you will be giving. And that will give you an idea that what you can think about that future, you can take up the avenues, what you go, as well as while going for your career, your career, you will be facing during interviews, exams also, that will also help you. As well as a researcher also you will come to know that this information will be very useful too. Sir has already told that if you give the seminar, that will be your knowledge sharing with other your colleagues, will benefit to them, others also, that you can look at. Another thing I would like to tell you that NBPGR, what we see is providing germplasm to the students also, PhD work. And so we get a lot of requests for that also. That also you can think of in your program while pursuing PhD, that you can avail the opportunity, this NBPGR, you can accordingly plan your program. Then I will say it's up to your hard work, learning, how much you can improve, how can think, and whatever you can interact later on with the scientists concerning of your interest if you have. Then not much. I think this is enough. That more will be dealt during the lectures. Sir has also explained you. And what I would like to say in the finally, there may be some shortcomings from our sides which you may find in our guest house and other facilities. So please bear with us for that. And I feel that your stage comfortable here. If anything, that you can tell to them. And I thanks to the organizers as well as my colleagues coming over here. And final thanks you will be giving. Okay. Thank you very much. Thanks a lot.
Thank you, sir. As we draw close to this session, I request Dr. Alawat to please felicitate the chief guest, Dr. K.C. Bunsel, with a token of our appreciation. Thank you, sir. Now, before we head for tea, it is an honor and privilege for me to propose the formal vote of thanks. First and foremost, we are highly thankful to Dr. K.C. Bunsel for accepting our invitation and agreeing to deliver this talk at such short notice. We are extremely grateful to the PI NAHEP CAAST, Dr. C. Vishwanathan, and the Associate Dean and Nodal Officer, NAHEP CAAST, Dr. K.M. Manjaya, for their constant guidance and support. We also express our gratitude to Dr. Rashmi Agarwal for her time and encouragement. We are greatly indebted to the Director, NBPGR, for all his support, especially for getting the NBPGR guest house ready on time. I am also very grateful to Dr. Veena Gupta for her guidance and support throughout the planning and preparation of this course. We are grateful to ICAR, NAHEP, Director and coordinators, and the CAAST team, IARI. A special word of appreciation to the HODs and OICs of various divisions and units of NBPGR for their support, and a special word of thank you to my officer in charge for understanding and allowing me to contribute time for this training. We are thankful to all the resource persons from within and outside the Institute for their contribution to the preparation of this training manual. Special words of appreciation and gratitude to all the members of various committees who have worked tirelessly to bring this training program together. Without your help, this training would not have been possible. We are especially thankful to the guest house in charge of NBPGR for putting in extra efforts to get the guest house ready well in time. We are also thankful to the entire staff and students of NBPGR who are directly and indirectly associated with this training program. And last but not the least, special thanks to all the participants, their university authorities, heads of divisions, and chairpersons for allowing them to attend this training program. Thank you, one and all.